we didn't have it. Just by looking at the CSET results, we could see that we didn't have BL37 in our isolates. We had some outbreaks with both clones and mixed isolates. So again, that's reassuring where you're seeing the same pattern within an outbreak, but again, you can see something else occurring there as well. And again, the only ones that matched an official race were some that matched BL35. We also did race testing on the spinach downy mildew samples that were sent in. I think actually we're only supposed to do the fungicide testing, but I think it gives a nicer picture if you have the race testing as well. We received fewer isolates, so there were 10 isolates here across a few outbreaks. And in this case, they appeared to match 3 designated races, race 1, race 2, and I think race 6. So, and this is the official race structure. You can see that this one is kind of questionable. We found it all to be positive. I think it's probably race 6, and they're all from the same outbreak in the southeast. Another notable one was this northwest outbreak that had 2 of this type and 2 of race 1. Okay, just moving on to the diagnostics, just got one more slide after this. So we were asked to develop and validate assays for Bremia. We developed a LAMP assay, which is just basically a simple PCR assay, which gives you a yes/no answer. It's not quantitative. And the thought was that that might be a simple assay that can be used in conjunction with spore trapping. There also existed a quantitative assay that tells you how many spores or DNA of the pathogen are in the sample that you're testing. So we also tested that existing assay. And we wanted to see whether the assays could be used for spore trapping and detection of Bremia inoculum in the field. So we did this with the help of a couple of commercial partners. The setup was that the commercial field had 2 traps. The traps were sampled twice weekly, and the samples were then tested using the quantitative test, which is sensitive and can detect low levels of the pathogen, and the yes/no assay, which is rapid and hopefully less prone to inhibition by dirt or other things in the sample. So I'm going to have to explain this slide quite carefully, I think. So the blue spots across the top are when spray applications were made during the season. The green line and the green dot are results from trap 1 tested using 2 different methods. Likewise, the black spot and the black line are results from trap 2 testing 2 different methods. So in theory, if both tests are equal, then every time there's a peak using one test, there should be a dot using the other test. So the reason we've represented them differently is because the line is quantitative. It shows you an amount of Bremia that's being detected, and the dot is just yes or no. But we do generally in this field see a correspondence between the dot and the peak, which is good news. You see a little bit of a lag sometime on one trap compared to the other. So for example, this higher peak seen on the 9th of August means that there was more DNA of Bremia detected in that sample. And indeed, a week after we saw that peak, disease was observed in the crop. So this is just an indication of what we're trying to do. We're still working on this technology with the help of the industry partners this year to see whether it can be useful in practice. It's not as simple as I may have suggested there, and we still need to work on this a little bit more to see it's of use in practice. So what are we doing this year? Well, Alicia Farmer's the AHDB PhD student. She's continuing to validate the markers to monitor the population. We still need as many Bremia isolates as possible this year, please. We're going to race test any samples provided this year, send the results back to people. We're going to continue with the in-field spore monitoring in commercial crops and with some other epidemiological studies on Bremia. Any spinach samples sent to us this year we will also race test, provided there aren't thousands, which I doubt there will be. But the ongoing race testing post 2022 is up for discussion. In the future, I think we could do that for the industry if that's something that they're required, but that would have to be a discussion, and that would then become chargeable in the future. Anybody who wants to talk about this or any other disease problems of lettuce or spinach, please just get in contact with me at that address, and we can chat about it. Okay, thank you.
Thank you very much, Alison. That was really, really good. That was a very useful summary of where we are with that work. Could I just ask, Alison, the report for CP184, will that be coming out shortly, or will that be available?
Yes, it will be. I think Tim's just bringing all the loose ends together at the moment for that report. Maybe he can comment on it later. But yes, it will be. It'll be on the HDB website, and I'm sure we can make it available as well.
That's great, thank you. We haven't really got time for individual questions, but there's a couple of questions in the chat, Alison. I don't know if you could perhaps have a look through and reply to them on the chat, if that's possible.
Yeah, I'll do that.
That's great, thank you very much. So from downy mildew, we're going to move on to spinach seed treatments. As you all know, the loss of thiram and following that metalaxyl has been a real problem. And so some work was started, AHDB funded, I believe, again, to look at spinach seed treatments. And we've got Catherine Eyre from ADAS. I believe she was working with Dave Kay at the time, no doubt she'll confirm that, who's going to talk to us on that subject now. Catherine, if you're able to screen share, please.
Okay, yeah, that's great. I will do. I'll just hopefully share the right screen. Can you hear me okay?
We can hear you here, yes, thank you.
Great.
See that?
Let me put it to presentation mode. Okay, great. Can you see the right screen? Does it look okay?
Yeah, it's in display rather than presenter mode, but I think it'll work.
So you can see the black screen. Is that, I can't tell what you can see. That's
No, that's fine. I would carry on with that, Catherine.
Okay, we'll crack on then. Fair enough. Okay, I just wanted to start by saying thank you very much for the opportunity to present this work. As you say, yes, Dave Kay, my colleague at ADAS, conducted and planned this trial last year, and he's now moved on to G. So I passed the baton on to me to do some of the data analysis and the presentation. And then there's also a contribution from Tim Pettit at the Eden Project, which will come at the end. So the overview, the aim of this project was to establish the efficacy and suitability of some seed treatments against damping off. We also had some biostimulant seed treatments that we wanted to have a look at and to look at their effects. And this was a really nice grower-led trial with demonstration trials based at multiple commercial sites across England. We used a summer variety which was susceptible to damping off in a single batch, trying to make it as uniform as possible across all the growers. And growers were asked to make multiple sowings during the season, and the seed treatments were done by Elson Seeds. So we had 8 treatments in all. 2 of those were conventional fungicides, 2 were biological fungicides, and then we had 3 biostimulants and physical priming. And you see it here at the bottom, this is the replication randomizations that were sent out to growers to ask people to, if they did multiple sowings, to put their treatments into different areas to mix it up a bit. So one of the things, we had 16 trials across England total. Most of the...
Hi, Catherine. We're still displaying only slide 1.
Oh, really? Oh,
yeah.
Maybe there's a bit of a lag.
Are you on your right slide?
Yes, I am.
Okay, must be us maybe.
Are you still on the overview?
No, slide 1.
Oh, that's strange. I'm
There is a cursor.
I'm sorry, what was
Yeah,
that?
I can only see slide 1 and 2 for some reason. It's not.
Catherine,
Okay.
potentially if you stop sharing and then share your main screen again. I think we're just seeing the PowerPoint slide rather than the
Okay.
presentation mode.
Let me see if I can try again. Okay. Can you see that now?
That
Yeah,
looks
much
better.
better.
Yeah. Yeah.
Okay. Now I can't advance my slides. Okay.
That's it. Yeah.
Okay, here we go. Good. Thank you very much. Okay, so here we are. So I guess this is what I've just talked about. I've got limited time, so I won't go over it again, but this was just to say the aims of the trial. Here are the 8 treatments. The yellow ones are the conventional fungicides. We have Maxim and a coded one. We have Integral Pro and another coded biological fungicide, and we have 3 biostimulants, and then we have physical priming. And this is the randomization schemes that were sent out to growers at the bottom. So we had 16 trials across. This figure at the bottom is just to sort of look at the timings of the trials and when the assessments were made. See that most of the trials were in a sort of mid-July drill date, and then we had some mid-August drill date for a second sowing. So the assessments were throughout the growing season. We tried to time these with sort of 50% target emergence, 100% emergence, and 100% emergence plus 10 to 15 days, and then again at harvest. And depending on sort of people's availability to do those assessments, we got readings from most of those time points, but not necessarily all for all trials. And each assessment wanted to look at sort of how many healthy seedlings emerged, how many damped off, and then look at what the percentage cover was across the whole plot, vigor and phytotoxicity. So first, after the seed treatments, ELSIMS did their standard germination trials, and they found that all treatments had some impact on germination by the final time points. Initial emergence at 7 days was better for most treatments compared to the untreated control. But in the end, AHDB 9848, and which was the conventional fungicide, and AHB 9763 had the most negative impact. The rest were sort of broadly comparable with the untreated control, maybe slightly less good, but I think that's something that you would expect with a seed treatment, to take a little bit of a hit on germination. Then we come to average percent damping off. Overall, the amount of disease was relatively low. You can see that it was only the maximum that we got was between 10 and 12%. And I think this was much reduced compared to the previous year in 2020, which was quite a bad year for damping off. I used the different quadrat sizes we used, so I converted the counts to a percentage damping off to make everything comparable between the different trials. And this first assessment is really the most important when those seedlings are first coming out after sort of about 7 days or so. Here, 9848, the conventional fungicide, and 9763 and priming did the best at the first assessment, but overall 9848 looked the best and performed across all of those time points. It's interesting that the Pythium sort of tends to affect earlier in the season, and Rhizoctonia tends to affect later. And we found that in other Scepter Plus studies that Integral Pro was more effective against Rhizoctonia than Pythium. So that's something just to think about when you look at these data about what sort of pathogens are at play, because it could be multiple different ones, and which actives or which products are most likely to take effect. So then we look at vigor with 10 being on a 0 to 10 scale, with 10 being the most vigorous and 0 being the least. 9732 was a biostimulant, and that was one of the most vigorous. I've put all the biostimulants here with dashed lines so you can see where they lie, and the untreated control is the very sort of fine dotted line. 9848 seemed to do quite well as well at the first time point. That's the light green one here, I think you can see. I guess vigor is always maybe perhaps a little bit of a trade-off with the treatments to protect against the disease. You may get slightly reduced vigor, but potentially better protection from disease. I think it's also at this point, it's good to say that, so, this is an unreplicated trial. So there's multiple different trials, they're in different areas, and so they haven't been statistically analysed. So all we can take from these data, and this applies to all of them, is that these are trends really, but there is some encouraging data here, I think. So then we look at average cover. As you might expect, you see increasing cover through time. Again, 9848 was the best of the conventionals and did slightly better than the untreated control. 9732 was the best of the bios, and 9734 was close behind. Priming also performs quite well in this particular metric, but Maxim 480 FS and 9733 were consistently the worst performers across all time points, and they really didn't do particularly well. That's the second and third from last bars. So when it comes to phytotoxicity, this is a bit of a tricky one to interpret in some ways, because it may have been interpreted differently by growers what phytotoxicity means. It's not necessarily the classic visible scorch symptoms that you might think of in other trials, more about sort of a impact on growth. And as that, so you wouldn't necessarily expect to see phytotoxicity in the untreated, but actually you can see that this dotted line, it sort of falls in the middle, and so it has been scored. So with that in mind, we need to be a little bit cautious about interpreting these data. But I would say that sort of it's quite clear at the top, this light green line is 9848, which seemed to have the least impact or the least phytotoxicity. Again, I should also say that the scale is slightly different here. So the higher the number, the less the damage or less phytotoxicity. So at the top is the best and at the bottom is the worst. So 9848 the best, and Maxim 480 is that sort of darker green line at the bottom. So then it comes to yield. We got yield data from one trial, which was great. Many growers reported that the crop was too thin to harvest at all, so we didn't manage to get any data for that. I don't know whether this is necessarily marketable yield, but it's interesting to see. But again, it shows us a trend, but we can't necessarily, as it's limited data, we can't necessarily apply it to. You need to be cautious about sort of believing it. But the best three performers I've highlighted in green here, we've got 9732, which was a biostimulant with 3.5 kilograms per linear meter. And then 9848, the conventional fungicide, also did pretty well, did much better than Maxim. And then another one of the biostimulants, 9763, also did quite well. And the rest, I would say, was, apart from priming, which was reduced, the rest were pretty much comparable with the untreated. So one of the other things that growers were asked to do was make comments about this, as well as taking sort of the numbers and the more sort of formal metrics. And this was really illuminating, and actually they aligned really well with the score data. And so in order to be able to try and sort of make sense of the comments and put them together, I coded the comments by how positive they were roughly, and gave them a sort of traffic light color. So red for bad, i.e. very poor, failure, green for good, and orange or amber for somewhere in between. And then collated all the comments from each grower for each treatment and each time point to see how they compared. And this is what I came up with. So these are the best ones, sort of mostly green, but with the odd bad one in there. So 9848 did pretty well, and priming also did very well. The untreated, unsurprisingly, did okay, but 9732, you can see it's a little bit of a mixed bag. Then we have the bad, as I like to call these ones, and these were the intermediate score treatments, which are a mixture of positive and negative, but mostly negative. And you can see lots of red and yellow and a tiny bit of green in there. You can see that the two biological fungicides fall into this group, Integral Pro and 9734, and one of the biostimulants, 9763. And then the ugly, the last two, these ones are predominantly had negative comments, and you can see that it's sort of things like very thin, failure, unable to harvest, poor, worst by far. And this included Maxim, the conventional fungicide, which seemed to perform quite poorly in many of the different metrics, and one of the biostimulants, 9733. So we're just going to switch gears a little bit now to look at some of the work that Tim Pettit at the Eden Project did, and this was to complement the field study by doing some more controlled bioassays. And he sort of used a pythium isolate that he isolated from the field to sort of do a grow through inoculation trial in soil, and then look at the treated different seeds and to look at see how efficacious they were in a more controlled environment. This slide is to sort of just say that when you're doing these sort of studies, it's very important to have the same amount of inoculum throughout your trials, but it's also the concentration is extremely important. You don't want to have so much inoculum that you sort of blow your seeds out of the water and overwhelm the protection that the seed treatments might give because it's too high. And it also wouldn't be representative of what these seeds might encounter in a natural environment, but also you want to challenge the seeds sufficiently so that you can see any differences and get a sort of a decent disease response. And this is always a challenge for any pathology trial, sort of finding the sweet spot. And Tim used two different concentrations of inoculum for his bioassay tests, 1.6 times 10 to the 3 colony forming units per gram, and then 2.2 times 10 to the 3, so relatively close. The first bioassay, there was very little disease. This was at the lower concentration. Overall emergence wasn't great. It was only about 68 to 75%, but you can see in the pictures that it was very similar across all the treatments. There was possibly some slight stimulation in 9763, a biostimulant, and 9848, the conventional biofungicide, and 9733, a biostimulant. So then he repeated the experiment in the second bioassay, but with the increased concentration level, and he increased it a little bit, not a huge amount, but actually it had a really great impact on disease manifestation, with emergence being less than 25%. And you can see that from looking at these trays. There's a lot of ones, the inoculated trays are the ones with these pink labels and where you can't see very much green at all. Probably somewhere in between these concentrations would probably be the sweet spot where you'd get about 50% disease. But there are some trends to be seen in this data, even though the numbers of emergence is quite low. Priming in AHDB 9848 gave better emergence than other treatments. And overall survival of the emergent seedlings wasn't high, even though some differences between some treatments. 9848 and 9733 had the greatest seedling survival, albeit relatively low. So just to summarize, I put all of those treatments which had notable performances, good and bad, in each category that we measured. And I've color-coded those so you can see where they pop up multiple times. And you can see that 9848 showed up as the best in most of the things, although in germination, it had a negative impact. But I think that sort of negative impact in germination was probably balanced by its superior performance in prevention of damping off and vigor and cover. And that was the conventional fungicide. 9732, which is in orange, was the best biostimulant and gave the best vigor and yield, good cover, but didn't necessarily protect against damping off. And 9763 was another biostimulant that was close behind. Important to say that these probably promoted growth rather than protected against Pythium directly. But it was a balance of finding those two things. So I think all in all, I would conclude that it was a really valuable trial, and there are definitely some products out there which could offer some protection against damping off to replace some of those products which have been lost. So I just want to say thank you very much for everyone who participated in the trial. I think it was a really nice trial. I'm happy I got to participate at the end. And thank you to Liz and Tim, seed suppliers and treaters, Dave, of course, and Grace and Kim at AHDB. Thank you.
Catherine, thank you very much. That was really useful, and it's very encouraging to see that there's some positive results there. I assume there'll be a final report. Is that out yet?
It's not out yet, but it's almost finished. I was waiting actually just to finish this, just to see if I could take on any comments to add into the discussion. But yeah, it will come out. Yes. Okay,
thank you very much. I think in terms of time, we don't have time to ask you questions directly, but could you have a look through the chat and just respond to any questions on your presentation?
Yeah. I apologize if I ran slightly.
No problem. Thank you ever so much, Alison. Sorry, Catherine. So we're moving on now to, we're staying with spinach and moving on to Tim Pettit. Tim's obviously been involved in both the preceding projects and was leading the downy mildew work. I think when he started the downy mildew work, he was at the University of Worcester, but he's now with the Eden Project learning. I think maybe has a university status. I don't know. Tim may be able to tell us. Anyway, Tim, if you're able to start sharing your screen, please, and we're on spinach downy mildew.
Okay, thank you very much. Can you folks hear me first of all?
Not yet.
You can't hear me?
Oh, we can hear you, but I can't see anything.
Yeah. Okay, yeah, sorry. I've been having a lot of fun with this system this morning, for one reason or another.
But we can hear you.
Can you see that?
Not yet.
What's this clever rascal doing? So I've got share. If I share window.
Ooh, something's happening. Aye.
Got it?
We've got your presentation. Can you go to presentation mode?
Where's
mode?
Sorry, say again.
If you can go to
you hear
presentation.
me?
I can hear you, yeah. We've gone again now.
I'm bringing to you my superpower of technical incompetence.
We'll get there.
So I go share screen. What did you say to do a presentation?
Share screen, and then when you get to your presentation, if you go to presentation mode, bottom right-hand corner of the PowerPoint.
Okay. Oh yeah, yeah. Okay, I'm with you. If it's working out, just shout bingo or something, so I can. Okay, they were in presentation mode.
You should.
You can see a full slide now.
Can you just go to slide 2 and see if it changes?
Yep, that's
no.
No,
so
Tim, I don't think we can see your presentation mode. So if you stop sharing and then reshare the kind of full screen with us.
Okay. It's lucky my talk's short, isn't it? Right. Share screen.
That's looking better.
Good.
That looks good. Thank you, Tim.
Okay. So this is going to be a fairly short presentation. Essentially, I'm going to share with you information that we've gained from the AHDB project CP184. So essentially, that's two sources of information. I'm not dipping into the stuff that Alison has dealt with, although of course she's a very important part of the CP184 project. So there's an element that I carried out, which was largely literature review. So finding out really what's been going on, especially in the US, in relation to this particular pathogen, but actually a range of downy mildews. So this was just one of a set of downy mildews that we were covering. Also looking into the prospects of potential for the future for decision support systems, so DSS. And then I'm going to briefly also cover Tom Wood's work, Tom Wood over at NIAB, who carried out some work looking at using PMA PCR to actually detect potential contamination of seed lots. So to start off with, just some general background about the pathogen. This particular downy mildew produces plentiful sexual resistant oospores. It's been demonstrated to have quite potent seed-borne infection. It's also been implicated to have soil-borne and debris infection. I think probably you can gain from my Pythium experience and phytophthora. I'm very much into soil-borne oomycetes, so this area interests me. I wonder how important that is within the overall epidemiology of this particular pathogen. It produces ample sporangia and conidia, and has also been strongly implicated in systemic infections. So it can actually cause infections, not necessarily immediately causing symptoms, and spread into parts such as seeds and the like, carryover infection. In common with most downy mildews, well, all downy mildews, Peronospora effusa likes cool and moist weather conditions. And there are a set of infection conditions that are stimulatory to disease, infection and disease. First of all, leaf wetness duration. These are all useful parameters I might add for developing disease forecasts or decision supports. So, for instance, leaf wetness duration is generally above 3 hours, and more recent work has shown that actually a minimum of 4 hours is required for infection of leaves by conidia, the asexual conidia spores. Conidial germination, maybe there certainly seems to be quite a large amount of variation within natural populations, and this seems to be reflected in the, if you like, the optimal range for conidial germination, which can range between 5 and 25 degrees C. There seems to be a kind of an optimum germination temperature of around 12, and then that tends to decrease with rising temperatures up to the 25. Survival of conidia, well, that's a little bit more vague, but it seems to run up to several days before they'll actually desiccate and potentially dry out. We have a latent period of a couple of days as well, and relative humidities that favor infection. Sorry, 7 to 10 days is the latent period, so the period between infection and actually symptom expression. And humidities at 90 or above stimulatory towards infection. So that sort of information could possibly lead to a development of models, but actually you can see there's quite broad ranges there, so it's actually quite a tricky one to pin down. But work has progressed in the US, developing a more direct approach in terms of measuring, just straight measuring of inoculum for a possible decision support. And in the USA, I say the rotarod spore traps. You can see this fella here on the right hand side here, rotating spore sampling surfaces. Those are sticky rods. Spores impact on those, get stuck, and then they can be analyzed. In this case, the guys are using quantitative PCR, so they're using a qPCR. This gives actually very sensitive detection, and actually DNA copy numbers have been found to match pretty much directly spore counts in a lot of cases. And the interesting thing is here that the numbers determined by qPCR on the traps 9 days prior to ratings could actually predict disease incidence. So you have if you can get the stuff analyzed quickly enough, you do actually have an early warning, potentially early warning system here. That combined with meteorological data, they're looking into actually developing this into a commercial service, and they're certainly in proto form already. They're also looking to improve DNA tests by using recombinase polymerase assays. So this is an ongoing thing. Seed-borne infection is quite an important route, potentially. The presence of Peronospora effusa oospores in seed was first demonstrated actually quite a long time ago, prior to 1980, but it was first really demonstrated quite conclusively in 1983. But it sort of was not given too much consideration until the sort of the late noughties, and then concentrated work on that led to viability tests using microscopy and plasmolysis, detecting viable oospores in 16% of tested seed lots. That was carried out in 2014. But one thing I would say about using microscopy and plasmolysis, I don't know how to say this politely, but it's one hell of a faff. You're looking at extraction and soaking these things in salt solutions and doing repeated microscopic analysis. And yeah, okay, when you look at the papers, you get some really nice shots, but actually a lot of the oospores are not tremendously convincing when you see them, whether they are sort of oospores sitting there nicely, not doing anything, or are they plasmolized? So actually, the discerning between viable and non-viable oospores takes quite a considerable skill and a lot of dexterity, and it's one hell of a faff. But concurrently with this, they carried out PCR tests, and again they detected Peronospora fusa inoculum quite well, very, very sensitive. And in this case, there were, as opposed to 16%, the concurrent test using PCR was picking up in 95% of the same seed lots. But the problem with this is it didn't actually, this particular approach, the PCR they were using, didn't actually give any indication of viability. And that kind of brings in the work, really introduces the work that Tom's been carrying out over at NIAD. Because what they've been looking at is detecting and quantifying viable downy mildew infections. They've been looking at leaves, but also very strong focus on seed-borne. And as I say, DNA diagnostics, very, very sensitive, can go down to single spores, as we're seeing with the qPCR. Problem is, generally can't determine between live and dead cells. And the danger there is you overestimate, potentially by quite a large margin, the level of risk, and it doesn't give you any measure of disinfection. So if you've disinfested seed by maybe a heat treatment or whatever, you don't really have any indication if you're just doing a straight PCR. So the idea with this was to develop viability PCR using Propidium monoazide, now PMA. A PMA is a photoreactive compound, but it can't penetrate live cell walls, so it can't get into viable cells. So when you treat a material that's got oospores in it, if they're viable, the PMA won't actually break into the cells. So if it does break into the non-viable cells, what it does is it will bind to the DNA within those dead cells, and that increases the number of PCR cycles in a qPCR. And what that means is that with the viable stuff having shorter, there's a lower number of cycles, means that you can actually discern between viable and non-viable DNA being detected in the sample. That's the theory at any rate. The procedures was originally developed for bacteria and has actually progressed quite well with the bacteria. The aim of this work was to test its utility for oomycetes, well, primarily mildews. So essentially, I'm not going to go through the essentially boiling this slide down. Essentially, what it was actually was successful with both Pythium effusa conidia and also with Pythium belbarii in basil. In that basically far fewer cycles are required to actually detect and quantify DNA in viable than in the non-viable material. So it was actually, it has been possible to actually test for viable spores in seed material, and this is now being progressed to further oomycete and broadening out to seed screens. And we'll be reporting on this in the final report for CP184, which has already been mentioned. So just to sum up briefly, we're talking about oospores, and there's some really nice shots out there. Unfortunately, I don't have any of my own, so I've had to sort of fill this whole thing with references to some of the really nice American work that's given some nice shots of Peronospora oospores germinating. These things are in seeds and seed coats, and that's okay. But and if seed-borne inoculum is the source of primary infections or primary inoculum in epidemics, with my Pythium head on here, I'm thinking, hello. If that's the case, is there a case for actually seeing if there is a link between this and the treatments we're looking for Pythium? Because if there's a bridge here that we could actually exploit to actually prevent primary infections, and it's working for one group of oomycetes, it seems to me interesting to actually explore whether seed treatments might also or seed coats might also contribute to restricting potential inoculum foci. Food for thought anyway, and that's where I'm going to conclude. Thank you very much. Cheers.
Tim, thank you very much. That was very interesting. If not a little worrying that these diseases may well be on the seed coat.
Yeah. Yeah.
Anyway, thank you very much for that.
Yeah.
Yeah, sorry, carry on.
Yeah, I'm going to say I'm not actually au fait with the exact numbers that that Tom's come up with, but yeah, that is certainly present viable in some seed lots, as far as I can gather.
Cool, thank you very much. Again, I think if you wouldn't mind just having a look through the questions on chat, and if answering the ones relevant to yourself, because I think we need to crack on to our next presentation. Thank you very much, Tim. So moving on, we're going away from diseases now and going towards aphids. We've got Dion Garrett, who's spent the last 3 years doing a PhD into the current lettuce aphid. I've no doubt most of you will have seen in the news the news that Cruiser has now been approved emergency-wise for sugar beet for this year because the aphid risk is deemed high, and aphid are predicted a month earlier than they were last year. Not saying that's current lettuce aphid, obviously peach potato aphid, but anyway, Tim, if you could stop sharing now, please, and Dion, if you could, I think that is your presentation. Yep. Yeah, Dion, if you're there, if we could move over to you, please.
Brilliant. Thanks, David. Can you see my screen? And I'm on the right one.
Yes, thank you. Yeah, that's good.
Brilliant, thank you. Yeah, in regards to the Myers persicki on sugar beet, yeah, the threshold for so Rothamsted have a forecast, and at the moment the threshold is 19% whether they'd be able to use Cruiser, and I think it's 68, so it's a little bit lower than 2020, which was really severe, but it could be quite a severe year this year as well. Just as a side note. So anyway, yeah, thanks, David. My presentation is literally just looking at the current lettuce aphid, Nassinovia rubus nigri. So it's the most damaging aphid on outdoor lettuce. And I'll just hopefully try to go to, there we go. So yeah, my PhD was, this is showing the last, culmination of 4 years of work. Really, I'm really going to just focus on the fieldwork side. It's quite multifaceted. It's very much, we don't know a lot about this aphid. There's a lot of unknowns. So this is what my PhD was funded for, is try to address a lot of these unknowns. So initially I looked at the long-term abundance, trying to forecast and improve trapping methodology, look at the alternative summer hosts and potential green bridges. And also did some molecular work as well, looking at the genome and studying the resistant breaking mechanisms of Nassinovia rubus nigri. And when I say the resistant breaking mechanisms, so Nassinovia is very difficult to control due to the nature of it infesting the center of the lettuce, so it renders a lot of foliar sprays completely ineffective and leads to a lot of customer rejections. So to combat this, in the 80s, there was a resistant lettuce variety which was introduced, which actually did confer complete resistance to Nassinovia, but unfortunately, I think it was in the early 2000s, it was actually able to break this resistance. So this was a single gene resistance, so it's not surprising really, but yeah, they managed to break the resistance, and now they're able to feed on it. So if some of you will probably know what this is, but this is a Rothamsted's suction trap, so they're 12.2 meters high. There's 16 currently distributed around the UK, and they use these to infer migration events of different aphids, such as Myers persicki with the sugar beet system. And really to illustrate the point of how cryptic this aphid is, So the aphids here are sort of highlighted in red, and this is Nassinovia rubus nigri. This is a very typical bulletin produced sort of in a given week. And you can see here we've got one in Dundee and one in Silwood Park. So it's very difficult to actually know when they're going to become a problem on lettuce. So what I first did, I looked at the long-term trends analysis of Nassinovia rubus nigri. So this is actually using all of the information of Nassinovia caught in the Rothamsted suction traps from 1965 to 2020. So this graph here essentially is the population trend. The orange line here is actually just shows any significant difference from year to year, and you can actually see that there's a difference in population density over time. So we can see it's significantly declined since 1965. So that's 76% decline since 1965, and the red arrow here is actually when the resistant cultivars were first introduced. So it does seem to have actually had an effect overall on the population of Nassinovia. I broke this down into region. So lettuce is primarily cultivated in the east. So that's the one I was really interested in. We can see here that, there's really big, significant declines from the start of recording up until sort of 2010, where the population's really plateaued. So I think it's reached a sort of a low threshold here, really. So yeah, I approached this project trying to improve trapping methodology. So I used these variety of traps here. So it's different trapping heights for the suction traps. So one of my hypotheses were Nassinovia is flying at lower levels, and as a result, we're not catching it in great numbers. So I used different height suction traps. I used malaise traps, which is this tented structure here to try to intercept their flight. I used different colored water traps. I used sticky traps. And yeah, over the combination of 2 years worth of work, over, I think there's essentially 10 months of trapping data, I caught very little Nassinovia. So yeah, my hypothesis was that, yeah, it was going to catch in higher numbers, and it didn't. So I rejected that. But we can see here that in terms of aphids, so this is the overall aphids that I caught during 2018, 2019, the suction traps, both 2 meters and 6, were much, much better than the other trapping methodologies, which is really interesting. But yeah, Nassinovia was very elusive. So I used a citizen science approach. So I put an ad on Twitter. I managed to get something in the RHS magazine as well. And essentially all these points on here is volunteers who grew lettuce on my behalf. They were able to get free lettuce, which was nice. So they got a summer's worth of lettuce, and I was hoping to get a summer's worth of data sent back to me. So the idea was that they got lettuce seeds, they cultivated them in their allotment or in their garden, and then any aphids they found on the lettuce they'd send back to me. So it's really nice coverage throughout the UK. I think it's about 160 odd volunteers in 2019. And this is the result. I got a lot of good pictures of lettuces, but I didn't get any Nassinovia. So despite that, it's still very elusive, and it really just reinforced the fact it's a very difficult aphid to find. So I got a good 100+ samples back. Mainly they're non-aphids. I got some ladybird larvae. I got some capsids, and then I got 21 different aphids. But no, I didn't get any Nassinovia. So I'm going on the assumption that sometimes a negative result is still a result, and it can be useful. So yeah, this is the typical life cycle of Nassinovia here. The green highlighted section here is when they become a problem on lettuce, but they actually start flying in early May. So they host alternate between a current, and then typically it's thought that they will then move on to lettuce and alternative hosts in the summer. But actually, they're flying in this sort of red period, but since they're not becoming a problem on the lettuce, I was like, one of my theories was, perhaps they're using an intermittent or a different summer host prior to migrating onto lettuce. So this is my second chapter. So I essentially used 4 different experiments. The first one was to look at field margins at Cambridgeshire. So this was at an organic site and a conventional site. So I did a lot of field margin surveys, trying to find them actually in the field margins and understand what species of plants are in the margins, and hopefully correlate that to the Nassinovia I found there. The second experiment was looking at the summer host plants. So instead of trying to actively search for them, I thought I'd try to actively attract them. So I planted different host plants in an attempt to attract them to some plots. The third experiment was looking at molecular tools to try to identify the plant species that had previously been feeding on prior to them being caught in any traps and on lettuce. And the fourth one was looking at the overwintering behaviours of Nassinovia. So the idea was these 4 experiments of this chapter essentially covered the whole life cycle to try to address some really fundamental questions that we didn't know the answers to. So my first experiment, looking at the field margins, it looks quite a complicated few graphs, but essentially on the left side, this is the organic farm, and then on the right, this is the conventional. And I did surveys in July, August, and September between 3 different typical habitat types. So that was a grass margin, semi-natural margins, and seeded mixes. So these are bird mixes and pollinator mixes. And the trend over the 3 months was, the number of species actually did decline during this time. So, we've got a notable drop in number of species throughout both grasses and broadleaf species. So that's actually quite interesting to know. And maybe there's something facilitating Nassinovia's movement onto lettuce later on in the year. Also did some field margin sweeps for Nassinovia. And surprisingly, I didn't get any Nassinovia. So they are there, obviously, but obviously just existing in very low numbers. My second experiment was looking at the plots. So I used a variety of known alternative hosts of Nassinovia. So this was mouse ear hawkweed, bird's-eyed speedwell, rough hawkbit, and chicory. I also used lettuce as a control. And I had 4 plots on the organic site dotted around the farm, and then 4 on the conventional. This was grown throughout May all the way through to October. And interestingly, I did actually manage to attract Nassinovia, but they were only found on the chicory, even despite the fact the lettuce was situated in the fields or in the plots throughout the time, but they didn't seem to feed on that. It was only just the chicory that they were attracted to and fed on. So that was an interesting find. Also, interestingly, the level of outbreak, this was during 2021. The outbreak was actually very low on lettuces as well. So they seem to be existing in very low numbers, and very close to lettuce, but weren't actually feeding on the lettuce. Third experiment, the concept was very simple. It's catching Nassinovia in the various traps, putting individual aphids in Eppendorfs, and essentially using some specifically designed primers to amplify the host plant barcode, and then sequence that, and then actually look at the results on a GenBank, a database, to actually see what plant species they'd been feeding on prior to them being caught. So the idea was to develop a method to actually identify what potential alternative hosts are, and therefore the green bridges onto lettuce. So the results I found from a preliminary experiment, I've able to amplify the host plant barcode within the aphid, which was brilliant, and this was up to 48 hours post feeding. So this was a laboratory experiment. So the proof of concept worked really well, and they also tested them on a few samples caught in the suction traps at Haney. As you saw, that the numbers were incredibly low, but the ones I did caught, they identified crested dog's tail and field forget-me-nots. What was really interesting about these two species, these species were actually found in the field margin surveys in experiment 1, but were actually senesced in sort of July, August time, which is actually the time when Nasonovia becomes a really big problem on lettuce. So this could actually indicate that these species are acting as a green bridge for Nasonovia's life cycle, because it only feeds on a very few select species of plants. And experiment 4, it was set up to understand the movement of Nasonovia from the winter host to the summer host lettuce. And despite the fact that lettuce was, these were semi-field experiments, they were caged, so they couldn't escape. The Nasonovia developed really nicely over winter on currants, and they started to produce winged aphids in the spring. The lettuce was there for them to move on to, but they actually didn't move on to it. They actually did seem to sort of disappear. So what I think happened is they essentially all died off. Their mortality rate was 100%, and there was just no suitable hosts for them to go on. So it does really prove, well, it did prove the idea that they need a secondary or an intermittent host prior to them moving onto lettuce. Very, very briefly, and really just to summarize some of the molecular work. So I looked at the resistant breaking mechanisms of the resistant lettuce. So the idea was to, I did some RNA sequence analysis, and essentially what all this graph is showing, it's a volcano plot, and all it's showing is the differentially expressed genes of individuals which are able to feed on the resistant lettuce and ones which are not. So the ones which are not are highlighted in blue, and the ones which are in red. So this is just comparing the different genes, and actually this particular gene here was highly and significantly differentially expressed. So this is some research I'm still currently doing some work on, but it really is a really nice breakthrough in terms of actually understanding what the potential mechanism that's involved in Nasonovia's ability to break the single gene resistance in lettuce. So that's a really nice find. And finally, I just did some population genetics analysis using microsatellite markers. So this is between 3 different year groups, so 2003 to 7, 08 to 14, and 15 to 20. And essentially what this shows is that Nasonovia doesn't really move between populations. There's a high level of inbreeding between all populations. So all these red dots here, this is essentially showing all the populations that I tested during these times. And there wasn't actually any movement between them, even between Wellesbourne and Rothamsted, which geographically are very close, but the populations are very distantly related. So it suggests that they are not moving they're existing in very low levels and not moving between populations. So I think this is one of the reasons why their populations are at such decline, I think due to the fact they are feeding on a very small number of host plants. And obviously with agricultural intensification has probably reduced their actual host plants, and therefore their overall population size is very low. So yeah, in conclusion, yeah, populations have significantly declined since 1965, even though they are still a big problem on lettuce. So I think we're seeing these sort of boom bust populations where their alternative host plants are senescing maybe early due to unfavorable weather conditions, which are forcing them onto lettuce. They have these big boom in their population, and then they're essentially just existing at very low levels. So despite the fact they're a huge problem in lettuce, I would regard them as a rare aphid, as in that they exist in low levels generally. And yeah, chicory could potentially be a nice avenue to invest the push-pull strategy for IPM. And yeah, investigating the single gene resistance in lettuce. And yeah, that is me.
Dion, thank you very much. That's fascinating. I find this whole subject absolutely fascinating, and I hope you've enjoyed your PhD. I know you've launched yourself at it really well, and it's I think you've got some really good learnings to come out of this. So thank you very much.
Thank you.
I think we're probably going to have to rename it the current chicory lettuce aphid, are we? The three-way?
Yeah, well, I think what is in a common name, really.
Right. Again, we're a little tight on time, Dion, so if you wouldn't mind just having a look through the chat and answering any questions from that point. I'm hoping that we'll still have you at the conference in October, because I think there's so much material there that it would still be useful. So I'm hoping that will still be there, but thank you very much indeed.
Thank you.
Thank you. So that brings us to the end of this webcast session. It's been a great session. We've had some really good learnings. I think some positives that I've taken away. It's really nice to see that the fungicides that we've tested on mildew are all really effective, some up to 100%. So obviously, the armory that we've got in these situations is looking good. It looks quite promising that we've got a seed treatment possibility for spinach that's quite effective. A bit disappointing that we're finding disease on spinach seed, but perhaps not to be unexpected. And some great work from Dion on his PhD. So it just remains me to thank the speakers, to thank AHDB for the funding into the first three topics, and of course the British Growers and BLSA for helping to organise this. So we're going to close the webinar now. This link will actually form into the BLSA AGM. So it's goodbye to non-members from here. Thank you very much. And it'll be over to Rob, I think. Yeah. People online that are joining the AGM, we're just going to have a 5-minute comfort break here and then we'll start the AGM. Thank you.